is just a really quick lecture over chromosomal mutations. You will need to know this for your exam. It's going to be really quick. So first, one example of chromosomal mutation is a deletion. And these ones we're talking about within a chromosome at a macro scale. So we talked about non-disjunction is if we had too many or not enough chromosomes. But now we're going to look at if there were large changes within a chromosome. And so a deletion is when a part of a chromosome is missing or deleted. So you can see over here, this chromosome originally had 4 colors, green, orange, blue, and purple, but the blue portion has now been deleted, and so you end up with a deletion within this chromosome. Next is duplication. This is a mutation involving the production of 1 or more copies of any piece of DNA, and so that means that there's a portion of the chromosome that's been duplicated. So here, once again, we have those 4 colors, but now you see that the blue has 2 copies of it in there, and so that blue copy has been duplicated in that example. The next one is insertion. This is a mutation involving the addition of a large portion of DNA into a chromosome. And so this is different than duplication, because in duplication it's just a repeat of something that was already in that chromosome, while in insertion it's an addition of a portion that wasn't originally there. So you see this white chromosome, now we have an addition of a gray portion which was originally part of a different chromosome. So a different part of a chromosome has been inserted into this one now. Then inversion is when a section of the chromosome is physically removed, flipped, and then reinserted. So here we had a chromosome that was A, B, C, D, E, F, G, H, I. D, E, F was physically taken out, flipped, and placed back in, so now it is F, E, D. So that portion is now backwards. That is called an inversion. And then the last one here is translocation. This is when a chromosome breaks and a portion of it attaches to a separate chromosome. This is different from crossing over because the chromosomes are not homologous. So if you remember, crossing over is between homologous chromosomes, and so if they switch, they're switching the same genes. It just might have different information within those genes. And so here we have 2 different chromosomes that are not homologous, and they end up switching portions. That's translocation, when it's not homologous and they end up switching portions.